So today we're going to be talking about the functional anatomy of prokaryotic.
Anybody here?
Yes.
Yes, I think there is some disconnection. It's a lot of rain and wind. Oh my gosh.
I hope it's
Yeah,
not gonna be happen during the exam.
that thunder is huge.
Yeah.
So we have to wait for 15 minutes, I think.
Yeah, but then
So
Yeah.
what if?
Yeah,
So do you guys know what if we have the same happen during the exam? What's gonna happen? He's gonna
no clue.
excuse.
He'll probably tell us or something.
Yeah.
Oh, okay.
He'll tell us next week before the exam.
So as I was saying, before I was so rudely disconnected, Wi-Fi has a problem sometimes. But you can all hear me now?
Yeah.
Yes.
Yes. Yes, Professor.
Yep.
Okay, so to start over again, we're going to be talking about prokaryotic cells and looking at the structures inside them and on the outside of them and doing some comparisons with eukaryotic cells. You spent a lot more time focusing on eukaryotic cells in 150, and so now we're going to spend a little more time on prokaryotic cells in micro. So of course we already know that prokaryote means there is no nucleus, and technically prokaryote means pre-nucleus, so before a nucleus, and then eukaryote means a true nucleus. Now prokaryotes are also missing all of the membrane-bound organelles like the Golgi, endoplasmic reticulum, mitochondria, chloroplasts. Those are not found in prokaryotic cells. So for some comparisons, prokaryotes just have one circular chromosome. And of course, it's not enclosed by the nuclear membrane. It's simply in the cytoplasm. But eukaryotes have those many long linear chromosomes surrounded by a nuclear membrane. You may recall that those long linear chromosomes are wrapped around histone proteins. That helps to organize those chromosomes to keep them from getting all tangled together. It helps them to condense during mitosis and meiosis. But prokaryotes just have that one circular chromosome, so it's not going to get tangled. So no histone proteins are found in prokaryotes. I just mentioned there's no membrane-bound organelles in prokaryotes. And most prokaryotes do have cell walls which contain at least some peptidoglycan. Eukaryotes, if they have a cell wall like plant cells, they have various polysaccharides in their cell wall. And prokaryotes reproduce by binary fission. That means they simply divide in two. But eukaryotes go through that complex process of mitosis and cell division. Sorry. So when it comes to the average size of most prokaryotes, they are small. Remember, we said we typically measure prokaryotes in micrometers. And so the average prokaryote, if it's a coccus, a sphere, is generally around 1 micrometer in diameter. Rod-shaped bacteria can be up to 8 to 10 micrometers long, but there are exceptions on both the high end and the low end. Now, the three most common shapes for bacteria are the coccus, and so the coccus is a sphere. And so we see the spherical bacteria. Some cocci are basically nice, perfect spheres. Others are a bit more squished, a bit more oval. When it comes to rods, the technical term for rod is bacillus. But we're not going to use that word to describe the shape. And the reason why is that bacillus is a proper genus, like Bacillus cereus. That's the genus and species of an organism. So not every rod-shaped bacterium is in the genus Bacillus. For example, E. coli. E. coli is a rod, but it's in the genus Escherichia, not Bacillus. So let's just use the term Bacillus only for the genus, and then we'll just use the word rod for the shape, and that minimizes confusion. Now, some rods are nice and long, so they're very obviously rods. Some other rods are actually short and fat, and so that does create a problem, because if you're looking at an organism that's shaped like this, It could be a coccus. It could be something like streptococcus or enterococcus, which is not perfectly spherical. It's more oval in shape. But it could also be a rod, like Klebsiella, which is a short, fat rod. So sometimes there is some ambiguity in terms of the shape. And so we sometimes use the term coccobacillus because sometimes we call it a coccus, sometimes we call it a rod. And, bacteria do whatever they want to do. They are what they are. Humans are the ones that try to define things. And not everything fits nicely and neatly into one category. So some rods can be short and fat, and some cocci can be oval, and they can look like the same thing. So that's a potential problem. And the other common shape is a spiral shape. Now there are some other shapes besides these three most common shapes. There are some prokaryotes that are star-shaped, like Stella. Looks like a star. Here we have a square-shaped or rectangular-shaped haloarcula. And there's even an archaean, that other major group are prokaryotes, which is kind of shaped like a football. And the example is Archaeoglobus. So there are some more unusual shapes out there. Those are not common. Those are not typical. And there's even another odd bacterium, which technically is a rod, but it's a curved rod. And they actually hook together using a specialized structure on the end of the cell. And so you get these, what they call rosettes. So again, not everything is either a rod, coccus, or spiral, and they're not perfect examples of those all the time either. Vibrio are curved rods. So they're not spirals, they're just curved, but they're rods. So not everything fits perfectly and neatly into every single category. So there are some variations. But for most bacteria, whatever shape they're supposed to have, they always have that shape. So they're monomorphic, one shape. Some bacteria, however, can change their shape and they're pleomorphic. A good example of that is the organism Corynebacterium xerosis. And I just mentioned this one because it's unusual, because it is pleomorphic. When it's young and healthy, it's a rod. But when it gets older, it gets rounder and looks like a coccus. So that is very confusing when you're making slides and trying to determine the shape of something. If you happen to grab a patch of bacteria that were old, they're going to look like a coccus. And if you get some young healthy ones, they're going to look like a rod. And so the student would tend to think they have two different organisms, a rod and a coccus mixed together. So there are some bacteria that can change their shape. And just to add something even more confusing, E. coli is a good example of a rod-shaped organism, but sometimes E. coli, so let's say that this is your typical E. coli. Sometimes when E. coli is dividing, a mistake happens, and the cell actually fails to divide. And so what you end up with is a really long E. coli. And these are very commonly found in our cultures. And we've seen them for decades. At first we thought there was some contaminant there, but it's just E. coli. that failed to divide and forms an unusually long cell. So not everything fits perfectly or looks the way it's supposed to look all the time. Now, some bacteria do stick together in characteristic arrangements, and the conversion didn't work well here. So if you have pairs of cells that are stuck together, we use the prefix diplo, as in diplococci or diplobacilli. If they occur in clusters, we use the term staphylococcus, because staphylo means clusters. So if you have clusters of cocci, they're staphylococcus. However, here we have another problem. Staphylococcus is a proper genus. Not every coccus that occurs in clusters is a member of the genus Staphylococcus. So you just have to be careful and say exactly what you mean and be very clear. So most cocci that are found in clusters are in the genus Staphylococcus, but not everything. A good example is Micrococcus. And Micrococcus luteus were those bright yellow colonies we saw in the environmental plates from yesterday. And Micrococcus luteus is a coccus that forms clusters of 4 cells. And we call these tetrads. So clusters of 4. And that's a very common characteristic you see in a Micrococcus culture when you make a smear. Now, you may see clusters of just 4 cells. You might see 8s, 12s, 16s, right? Multiples of 4. And this is a predominant characteristic if you're looking at a slide. You see lots of clusters of 4, but the clusters of 4 may be stuck to other clusters of 4. Now, in the Staphylococcus culture, when you make a smear and look at it, you will occasionally find a random cluster of 4, but it's not a predominant characteristic, right? Just like you'll see some random pairs in a Staphylococcus culture or a Micrococcus culture. It's not a predominant characteristic. It's just that, random things happen. So when it comes to the arrangement, you don't want to get too concerned about the arrangement, because while you always look for the predominant arrangement, you may see a variety of other things in the smear. And another factor that affects the arrangement is how thick your smear is when you make it. If it's too thick, all the cells are stuck together and you can't actually see the characteristic arrangement. And if you subculture and maintain the same bacteria, and you subculture month after month after month, they actually tend to lose their characteristic arrangement. So the best way to see a characteristic arrangement on a slide is to look at what's called the wild type version, something isolated from a patient or the environment, or a brand new fresh culture that you just got in the mail. And then you'll see that characteristic arrangement. Now, some cells occur in chains. So the prefix there is strepto. But once again, we have to be careful. Streptococcus is a proper genus. So just because you have chains of cocci does not necessarily mean they're in the genus Streptococcus. A good example, Enterococcus. Enterococcus actually used to be classified as Streptococcus, but it turns out there's not many chains in the Enterococcus cultures. So they really didn't fit into the genus Streptococcus. So they're now Enterococcus. So when you look at a smear of Enterococcus, you'll see some small chains here and there, and you'll see some random clusters, right? There's no particular arrangement other than to say that it's clustered. But if you said, I have Staphylococcus, what does that mean? You have members of the genus Staphylococcus, because it could be Enterococcus, it could be Micrococcus, right? It could be a lot of things. So just when it comes to this nomenclature, you do have to be careful and really think about what you want to say and what you mean. And chains of rods would be streptobacilli. Yeah, question about him?
Yes, Professor. So, based on the arrangement, we cannot tell the actual bacteria, right?
Well, I could tell based on the organisms we commonly use in lab. You could not tell because you don't have the experience yet, but in general, just looking at a stained slide, you cannot conclusively identify the organism just looking at it. If you saw clusters of 4 as a predominant arrangement, that strongly suggests it's the genus Micrococcus. You don't know for sure, but it does strongly suggest that.
Okay, thank you.
So many bacteria have sticky structures or sticky coatings on the outside of them. And we collectively call these sticky coatings the glycocalyx, because glyco means sugar and calyx means a covering, so a sugary covering, and sugar is sticky, right? And we saw a capsule yesterday when we looked at an example of a negative stain, and we'll see that again later on in a lab next week. But a capsule is normally a thick, well-organized layer of polysaccharide. So it's thick, it's organized. It is sticky, and it also serves a protective function for those bacteria that have a capsule. One of the things that the immune system tries to do is to eat bacteria using a cell called a phagocyte. So phagocytosis means to eat. And so cells protected by capsules, it's very hard for a phagocyte to eat it. It just doesn't like that polysaccharide layer. So if a pathogen is surrounded by a capsule, it's a much more dangerous pathogen because it's very hard for your immune system to fight and destroy that pathogen if it's surrounded by a capsule. Now, slime layers are also sticky, right? They're polysaccharide, well, they're carbohydrate layers, but they're not as well organized. So they're thinner, not as well organized. A good example, let's say you buy a packaged lunch meat like bologna or sliced ham or something like that. And you open up the package and you take out a few slices and put it back in the fridge and kind of forget about it. And maybe a month later, you go to grab a slice and it's all slimy. It's because now it's covered with bacteria and they're producing these slime layers. And so it feels very slimy. But because these structures are sticky, they do help bacteria to attach to each other as well as to attach to substrates. And that can also enhance the pathogenicity of a bacterium because it's able to stick to your cells and tissues. Now, flagella are protein structures used for movement. And the technical term is motility with a T. And we're not going to go into this, but flagella are actually very fascinating molecular motors because they have to move that flagellum back and forth. That requires ATP and a special type of motor to do that. And so eukaryotes have flagella as well. They're much larger than prokaryotic flagella, and they're made of a different protein. but the exact mechanism in terms of how it works is the same. When it comes to prokaryotes, their flagella are made of the protein flagellin. These are typically anchored to the cell wall. And like I said, they have a fascinating molecular motor, which allows the flagella to whip around. And that will move the bacteria. And sometimes bacteria do want to move. They want to be able to swim. Now, if flagella are present, there are 4 characteristic arrangements. One single flagellum, we call that monotrichous. And trichous is a Latin word for hair. So it's like one single hair. A flagellum on each end is amphitrichous. Clusters of flagella are lophotrichous. It's usually just found on one end, but sometimes you can have more than one cluster. But clusters are lophotrichous, and if the bacterial cell is covered with flagella, we call that peritrichous, because peri means all over or surrounding. Remember that flagella are teeny tiny. The only way to see them, other than an electron microscope, is to use a special stain, which makes them thicker. And that enables us to see them on a prepared slide. Not for real, not live action. Unfortunately, we're not going to be able to do the actual lab motility face-to-face, and you would have the pleasure of trying to find and focus these live, unstained bacteria, right? They're not stained, right? Because the stains kill them, and we're trying to observe motility, so you would have to actually find them and focus them and then watch them and try to determine from their movement if they were actually motile. And that's very challenging to do. But motility with a T is the movement of bacteria, and it's a purposeful movement. Bending, twisting, twirling, rotating, or actually swimming in a lateral line, so swimming directionally. If you have bacteria that are swimming directionally, that's really easy to tell, right, that they're motile. But all particles suspended in water move. And we call that kind of movement Brownian movement. So all particles suspended in water will randomly vibrate because the water molecules are hitting those particles and causing them to randomly vibrate. And the same thing happens with bacteria suspended in water. They will randomly vibrate like crazy. Sometimes it really looks like they're motile, when in fact they're not. They're just randomly vibrating. So trying to determine true motility from Brownian movement can be challenging if you're just looking at live unstained bacteria. Now, movement either towards or away from a stimulus is called taxis. So not taxi in this case, or taxis, but taxis. So why would bacteria want to move towards something? Well, if they're moving towards food, that is chemotaxis. If it's a photosynthetic prokaryote and it's swimming towards the light, that would be phototaxis. Not all bacteria have flagella, so not all bacteria are motile. Some are, some aren't. Even within the same species, flagella can be variable, right? So they may not always be produced. Now, because the prokaryotes have these special proteins in their flagella, they're not all exactly the same. So the protein flagellin can have some slight differences, and we can actually use those differences to type different strains of bacteria. So a good example we see here is E. coli 0157H7. So H is the type of flagellar protein that they have. So there are many different types, but we can use the type of flagellar protein in order to type a different strain of the same species. So E. coli, there are many different species, or many different strains of E. coli. In general, E. coli is a very good and beneficial microorganism. Our intestines are full of it. It's very beneficial. Not 0157H7. That's a nasty E. coli. That's a pathogen. It causes severe diarrhea. And it can be fatal if you're really young or really old or you're immunocompromised. So even normally very beneficial bacteria may have harmful strains. So it would be wonderful if the flagella were large enough to actually see. They are not. So the cartoons and animations that are often shown are not very realistic because we can't actually see those flagella, but in general, you try to determine if there's some purposeful type of movement. As I said before, bending, twisting, twirling, spinning, or actually swimming directionally from right to left, left to right, top to bottom, something like that. So we have some videos and animations that I'll show you. So I have to share my screen for this so we can see it. Let me turn down. Okay, so can we all see chemotaxis here?
Yes.
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 Motile bacteria and archaea generally swim using rotary flagella, which are helical propellers that drive the cell forward. E. coli bacteria have 2 to 6 flagella randomly distributed around the cell. During an uninterrupted run, in which the cell moves steadily forward, all the flagella spin counterclockwise as a bundle. Periodically, one or more flagella reverse their rotation to clockwise, disrupting the entire bundle and causing the cell to tumble. The cell ends up pointed in a random direction. Soon, all of the flagella again rotate in the counterclockwise direction, and the cell swims forward. The repeated course of swimming and tumbling results in what is known as a random walk. This video shows E. coli bacteria swimming and tumbling, reorienting and swimming again. Most flagellated cells have an elaborate sensory system that enables them to swim toward favorable environments that contain attractants, such as nutrients, and away from inferior environments that contain repellent signals, such as waste products. This sensory system is known as chemotaxis. In E. coli, the flagella bundle together behind one pole of the cell, while the opposite pole contains a diversity of chemotactic receptors. When the cell moves away from an attractant, that is, down the concentration gradient of the attractant, the receptors send signals that trigger one or more flagella to rotate clockwise. The bundle flies apart, causing the cell to tumble and then swim in a new, random direction. As the concentration of an attractant increases, the cell generates fewer signals to switch flagellar rotation, and the cell tumbles less often. The pattern of movement in chemotaxis is called a biased random walk, in which the cell tends to migrate toward the attractant. In contrast, a random walk has more frequent changes in direction and shorter runs.
And then we also just learned how bad closed captioning is in YouTube. So I have some other videos of motility using the protozoa. So these are much larger eukaryotic organisms. And when it comes to protozoa, some protozoa may have flagella, others cilia, and still others pseudopods. And so we're going to look at paramecia, euglena, and amoeba. And we're starting off with paramecia. And there's no sound on this. And so paramecia have cilia. If you look on the edge of the cell, you can see little cilia on the edge. Now this paramecium has been slowed down, or the video has been slowed down, because normally they zip around. Yeah, here, it's showing you at 50% normal speed, because normally they actually move pretty fast, and it's hard to keep them in focus. This is euglena. It's just one. It squishes up and out because it's in a thick solution of methylcellulose, which is there to slow it down. Otherwise, it'd be moving way too fast. And you can see one flagellum on the end. It's not moving because it's in a solution that's too thick. It's like you trying to swim through molasses or syrup. And here we see amoeba. And amoeba are protozoa which kind of crawl around on surfaces and substrates. And they extend what are called pseudopods. So they extend their plasma membrane in one direction and then shift to all the cytoplasm and organelles, which are what you see inside of it right now, in that direction. And this has been sped up considerably, right? This is real time, right? They're pretty slow. But notice all those little things inside are the organelles. And amoeba are especially cool to watch with a phase contrast microscope because they're all shiny and shimmery and glittery. So we already saw that, so just stop. And it never takes me back to where I left off, so I always have to jump ahead to get back to where we were. Now there are some spiral-shaped bacteria, the spirochetes, and spirochetes also have flagella, but they have what are called endoflagella because they don't stick out. They actually run along the cell body. And they also call the endoflagella axial filaments. And so the way it works is that the axial filaments are attached to one end of the cell. And then the axial filaments run along the cell body, but there's another outer covering of the cell called a sheath. And so the axial filaments, those endoflagella, move in between the cell body and the sheath. And because they're anchored or attached to one end of the cell, as they're freely moving on the other end, it causes the entire cell to twist like a corkscrew. And so these are pretty cool to watch because they do tend to spiral like a corkscrew. And again, I should have showed you this before. There's a little short YouTube video. I'll just show you some spirochetes wiggling around. And these are actually the spirochetes that cause Lyme disease, which we'll talk about a little bit later. You can see them kind of wiggling around and actually moving directionally because they're twisting. So clearly they're motile. Near the end, you'll see another little bacterial cell swim into the field of view. There we have this other little guy here, which is also motile. So there are many spirochetes that seem to be pathogens. So I'm not sure why exactly so many spirochetes are pathogens, but we mentioned one, Treponema pallidum, which is also a spirochete. Borrelia, which causes Lyme disease, is a spirochete. And one time, a long time ago, I had a student, and he had some white stuff growing on his tonsils. And so he was curious, and he wanted to take a look at it. So I gave him a sterile swab, and he took a sample and put it on a slide, like a wet mount. And he had the most amazing spirochetes I have ever seen. Just a gazillion of them wiggling around all over the place. And I told him, this is not normal. You need to go see a doctor, right? Because you shouldn't have that going on. But they were really amazing to see these living spirochetes, and just thousands of them wiggling around like crazy. Fimbriae are protein structures which are sticky. And so here we see a bunch of fimbriae surrounding this bacterial cell. They also function in attachment, and they can help the bacteria stick to each other as well as to substrates like your cells and tissues. Pili, and that's the plural. The singular is pilus, and cells that have a pilus, they only have one. And we'll talk about the pilus much later on when we talk about bacterial genetics, because the pilus is a special structure used for attachment, and one cell uses it to attach to another cell so that DNA can be transferred from cell to the other. Most bacterial cells do have a cell wall. Not all of them do, although almost all of them do. And that cell wall helps to protect those cells from osmotic lysis, because most of the time, bacteria are growing in hypotonic environments, or at least less hypertonic than the inside of the cell. So water does have a tendency to move into the bacterial cells, and the cell wall helps to protect them from bursting. Most bacterial cell walls are made of peptidoglycan. Some cells, like the gram-positive cells, have a lot of peptidoglycan. The gram-negative cells have a little bit. So what is peptidoglycan? Well, the name should give you a clue. Peptido as in peptides, and glycan as in sugar. So peptidoglycan is made of polypeptides and disaccharides. And the two disaccharides are N-acetylglucosamine and N-acetylmuramic acid, which you do not need to memorize, but they are linked together by these polypeptides. And peptidoglycan is actually structurally similar to chitin, which is found in insect exoskeletons, as well as the cell walls of fungi. But peptidoglycan is found in most prokaryotes, well, let me back on that. It's found in bacteria. the archaea, other prokaryotes, some of them have cell walls, but they're not made of peptidoglycan. So now we're going to learn the difference between a gram-positive cell wall and a gram-negative cell wall and see why the gram stain works the way it does. So for a gram-positive cell, the key point is that it has a very thick layer of peptidoglycan. It's very thick. Gram-negative cells have a very thin layer of peptidoglycan and a second membrane. So gram-negative bacteria actually have two lipid bilayer membranes. In between them is a thin layer of peptidoglycan. So those are the key points. Thick peptidoglycan in gram-positive cell walls, very thin peptidoglycan in gram-negatives, plus an extra membrane surrounding the gram-negative cell. As we'll see, that gram-negative membrane does provide some protection to the gram-negative bacteria. Now, when it comes to acid-fast bacteria, and I mentioned Mycobacterium last time. Oops. As in Mycobacterium tuberculosis or TB. Mycobacterium has a special cell wall. It's thick and waxy, and the waxy substances are mycolic acids. So mycobacteria technically do not have enough peptidoglycan to be classified as gram-positive bacteria. However, when you gram stain them in lab, they're typically purple. So they look gram-positive when you do a gram stain, even though technically they're not gram-positive bacteria because there's a minimum amount of peptidoglycan required to be classified as gram-positives. And they have less than that. They have more than gram-negative bacteria, though. And that's why Mycobacterium is not a gram-negative organism. It's not gram-negative, it's not gram-positive. It's kind of in between because it doesn't have enough peptidoglycan to be gram-positive, but it's got too much to be gram-negative. So here we see the gram-positive cell wall. Again, the key point is that very thick layer of peptidoglycan sitting on top of the cell membrane. We contrast that with the gram-negative, very thin layer of peptidoglycan sandwiched in between two membranes. So gram-negatives have a double membrane. As I said, that double membrane provides some protection from the immune system as well as from certain antibiotics. But that outer membrane is also somewhat toxic. So you see the lipopolysaccharide here. In particular, the lipid A is actually a toxic substance. It's only toxic when the gram-negative bacteria die and break apart and they release the lipid A, but fortunately it's not very toxic. And as long as the bacterial cells are intact, then there's no toxicity from the lipid A. Now the O polysaccharide that you see there, that is something that we can use to type different strains of bacteria. Remember the E. coli O157. The O is the O polysaccharide, at number 157. So we can use the O polysaccharides to type different strains of bacteria. So let's focus on the gram-negative. So there is a space in between the inner and outer lipid membrane. It's called the periplasm. That extra lipid membrane also helps to protect those bacteria from phagocytosis. It's not as good as a capsule, but it can help to prevent phagocytosis. Certain antibiotics either cannot penetrate to membranes, right? So the antibiotic can't get through to kill the bacteria. Or if the antibiotic, like penicillin, which targets peptidoglycan synthesis, has no effect on gram-negatives because they don't need much peptidoglycan. But they got two membranes and just a little bit of peptidoglycan to begin with. So that's why the penicillins are not effective against gram-negative bacteria, is because they don't care. They don't have much of a requirement for peptidoglycan anyway. But gram-positives, they need that thick layer of peptidoglycan. And so that's why penicillin disrupts the peptidoglycan. When it comes to complement proteins, when we get to the immune system, we'll learn that complement proteins are one of our non-specific defenses, which means they work against a wide variety of pathogens. And they're basically proteins floating around in the blood serum. When properly activated, these complement proteins assemble and poke holes through cell membranes. The problem with the gram-negatives is that they do have to poke holes through two membranes, which they can do. It's just harder. I mentioned the O polysaccharide is antigenic, so there are different types of it, so we can type different strains of bacteria. Lipid A is an endotoxin. Endo just means it's a component of the cell, and it's not really very toxic. Of course, there are protein channels through the membranes to allow substances to move to the inside and outside of the cell. So, any questions on the difference between gram-positive and gram-negative cell walls? So, why does the gram stain reaction work the way it does? Well, remember, the primary stain is crystal violet. You put that on your smear for a minute, rinse it off, then you add iodine and leave the iodine on for a minute. Iodine forms a complex with the crystal violet, which sticks to peptidoglycan. And then when we decolorize with the 95% ethyl alcohol, that helps to denature the proteins in the peptidoglycan and helps to trap that crystal violet. Since gram-negative bacteria have very little peptidoglycan, there's nothing for the crystal violet iodine complex to stick to. So when you use the 95% ethyl alcohol, it dissolves away the outer lipid membrane and it washes away all the crystal violet. So that's why the gram reaction works the way it does. That crystal violet iodine complex sticks to peptidoglycan. Gram-positive cells have a lot of peptidoglycan, and so the crystal violet sticks. The gram-negative cells have very little peptidoglycan, so the crystal violet cannot stick. It all washes away. Now, last time I mentioned one of the problems when gram staining, and one of those problems was that old gram-positive bacteria can stain pink or red, so they look gram-negative. That's because old gram-positive bacteria, their cell wall begins to break down. So the peptidoglycan begins to break down, which means it's harder for the crystal violet to stick, right, to those broken down gram-positive cell walls. So all the crystal violet may be washed away with the alcohol. And so the cells stain, red or pink and look like they're gram-negative. So we'll talk more about problems with gram staining next week. So I did say that most bacteria have cell walls, but not all of them. Mycoplasma, not to be confused with Mycobacterium, two completely different organisms. Mycobacterium has a very thick, waxy cell wall. Mycoplasma has no cell wall, no peptidoglycan, so they have no definite shape. They're pleomorphic, right? Because they have no cell wall, they're not rigid. So why don't they explode, in a hypotonic environment? Well, they may, but they also have sterols in the plasma membrane. And you remember from 150, sterols like cholesterol can help add rigidity to the lipid membrane. And so sterols provide some structural integrity to the mycoplasma cell membrane. When it comes to the archaea, that other major group of prokaryotes, some of them may not have a cell wall. Many of them do, but the cell wall does not contain peptidoglycan. It contains other things, which you don't need to know. But the archaea, that other major group of prokaryotes, do not contain peptidoglycan. So that was one of a few significant differences, and one of the reasons why the archaea were separated from the bacteria. Because remember, the two domains of life that are prokaryotic are bacteria and archaea. And they're two separate domains. So, if we damage the bacterial cell wall, we can kill the bacteria, right? So we can damage the cell wall, we can kill those bacteria, and there are many ways to do that. One way is a lysozyme, an enzyme that helps to break down peptidoglycan. Lysozyme is one of our non-specific defenses. You have a lysozyme all throughout your body. It's in tissue fluids, it's in saliva, it's in your tears. And so lysozyme helps to break down peptidoglycan and kill bacteria, because now water can move in and the cells will explode. Interestingly, when we learn about bacterial viruses, the way that bacterial viruses penetrate the bacterial cell wall is by using the enzyme lysozyme. So that's a very interesting circumstance where animals produce lots of lysozyme to help defend against bacteria, but these viruses somehow evolved or acquired the enzyme lysozyme, which allows them to penetrate the bacterial cell wall and infect bacteria. So it's pretty interesting, coincidence. We talked about penicillin. It targets gram-positive bacteria because it interferes with peptidoglycan synthesis. And gram-positives are extremely dependent on that thick layer of peptidoglycan. Not so much for gram-negatives, which is why penicillin is not used to treat gram-negative infections. When it comes to the words protoplast and spheroplast, don't worry about any other definitions you see, because it depends on what kind of organism you're talking about. Are you talking about a eukaryotic cell or a prokaryotic cell, like a plant cell that's lost its cell wall? And so don't worry about the exact definitions of protoplast and spheroplast, because it kind of depends on what source you're looking at and what cell type they're talking about. All you need to know with protoplast and spheroplast is that either all or parts of the cell wall has been damaged or destroyed. L forms are bacteria that used to have a cell wall but no longer do. Right? So for some reason they lost their cell wall, which means they will swell up with water, which may or may not cause them to explode. But whatever the case, if we damage or destroy the cell wall, those bacteria become susceptible to osmotic lysis. Water will move into the cell and the cell will explode because there's no cell wall to protect them anymore. We also have certain detergents and chemicals which also damage the cell wall and make the bacteria susceptible to osmotic lysis. So we talk about disinfectants and antiseptics. We'll talk about some of those other chemicals. So we've mentioned a lipid bilayer membrane, so we got to review some things, which is all just stuff you've already learned in Biology 150, so nothing new here. But you know that the lipid membrane is made of phospholipids. And of course, a phospholipid has a head and tails. What's special about the head?
Hydrophilic.
It's hydrophilic, right? And that's because there are some charges associated with the phosphate and the choline that's in the head structure. And because it's charged, it likes to interact with water. Because remember, water has those polar covalent bonds, so the partial positive and negative charges on the hydrogens and oxygens. And the fatty acid tails are hydrophobic. They hate water. And the reason why is the tails are all hydrocarbon. They're oily. And we know that oil and water do not mix, right? They don't like each other. So if you mix phospholipids in a beaker of water, they will automatically form a bilayer configuration. And that is so the hydrophilic heads can face the aqueous environment, the watery environment, and the hydrophobic tails can face each other. And that is the most thermodynamically favorable configuration of phospholipids in water, is they form a bilayer. That bilayer is selectively permeable. Certain things can dissolve through the bilayer, other things cannot. But remember, very charged molecules cannot dissolve through the layer. Large molecules cannot dissolve through the layer. They need transport proteins to allow them to move through. So the cell membrane is another target, because if we can poke holes in the membrane, the cells will leak and they'll die. Water will rush in, but mostly things leak. So alcohol will dissolve lipids, whether it's bacteria, whether it's your liver cells or your brain cells. Alcohol dissolves lipids. The quaternary ammonium compounds, or the quats, like benzalkonium chloride, really does a lot of damage to the cell membrane. Certain antibiotics, like the polymyxins, poke holes through the cell membranes. So again, if the membranes are full of holes, the cells will leak and they will die. So we can destroy bacteria by damaging their cell walls or damaging their cell membranes. So let's review osmosis and diffusion, hypertonic, hypotonic, and isotonic. We know that a hypertonic solution has a high concentration of solutes, and the most common solutes are salts, sugars, even proteins can be solutes. But the solute is dissolved in the solvent, which in this case is water. So a hypertonic solution has a high concentration of solute. A hypotonic solution has a low concentration of solute. And remember, these are relative to the other side of the membrane. Isotonic is the same concentration of solute on both sides of the membrane. So if you put a cell, like a red blood cell, into a hypertonic solution, where does the water go? What happens to the cell?
Shrink.
It expands.
The cell will shrink. It'll shrivel, right? Because water leaves the cell. And just remember, water always moves into the hypertonic solution. So water moves into the hypertonic environment. If we put a red blood cell into a hypotonic solution, like fresh water, what happens to that red blood cell?
It will inflate.
Well,
Burst.
it'll explode, right? It'll fill with
Okay.
water and burst, right? Because a red blood cell just has a membrane, no cell wall. And in an isotonic solution, animal cells are happy, right? There's no net movement of water. Of course, with plant cells and prokaryotic cells, some slight differences. When it comes to prokaryotes, they are happy in isotonic solutions. They can handle a somewhat hypotonic environment, at least for a while, because they have a cell wall. But if you damage that cell wall, the cells will explode. And then in a hypertonic environment, like a high salt solution or high sugar solution, it sucks all the water out of the cell. But the cell doesn't really shrink because it's got a cell wall. What happens is the cell membrane pulls away from the cell wall, and the inside of the cell shrinks. And we call that plasmolysis or plasmolysis. That may or may not kill that bacterial cell. It may just put it into suspended animation. So it goes dormant and waits until favorable conditions return. But many bacteria have no problem in pure water unless you damage their cell wall. And many bacteria can survive relatively hypertonic environments for a while as well. So bacteria are very adaptable. So the cytoplasm is everything within the cell membrane. In 150, you probably use the term cytosol more, right? So you remember the term cytosol. So the cytosol was the liquid part, not including the membrane-bound organelles, so not including the nucleus or the Golgi or the ER or the mitochondria. It's the watery part in between all of those organelles, whereas technically the cytoplasm includes everything within the plasma membrane. Since prokaryotes don't have any of the membrane-bound organelles, we just call the inside the cytoplasm, not the cytosol. Now, there is no nucleus inside of a prokaryotic cell, but the chromosome does tend to be concentrated in one particular region. And so they sometimes call that the nuclear area or the nucleoid, but there is no nuclear membrane. It's just the chromosome does tend to be concentrated within a region inside of the prokaryotic cell. So we're going to finish up by talking about a very important inner structure that some bacteria can produce. Not all of them, but only some can. We'll talk about this next week in lab. But endospores or spores are survival structures produced by some bacteria. The two major groups of spore-forming bacteria are the genus Bacillus and Clostridium. They're not the only ones that can produce spores, but they are the major ones. So a spore is a dormant structure. It's a resting cell. There's no metabolism, no transcription, no translation, no nothing. It's completely dormant. Because it's dormant, it's highly resistant. There is a very thick type of cell wall called a spore coat. It's very thick, it's very tough. So spores are highly resistant to the environment. Desiccation or drying, spores require absolutely no moisture to survive, none. Heat, in order to kill a spore, you have to boil it at least 30 minutes in order to kill it. One of the reasons why John Needham's experiment failed is that he most likely did not boil his broth at least 30 minutes and kill everything like Pasteur did. Many chemicals have no effect because what chemicals do is interfere with metabolism or DNA replication or transcription or translation, something. None of that is happening in a spore. It's completely dormant. Radiation. Radiation works by causing so much damage to DNA that when the cell tries to replicate and divide, it can't. There's too much damage. But since it's dormant, radiation has very little effect on spores. And they've actually sent spores up on the space shuttle and exposed them to outer space. And it depends on what kind of organism it is, but they can survive for certain periods of time. And speaking of time, spores can survive an extremely long period of time. Exactly how long, they don't know. Hundreds of years, maybe thousands. Some speculate millions, but DNA is not stable enough really to last that long unless it's preserved in some way. But spores do last a very long time. So we do find spores in soil because both Bacillus and Clostridium live in soil. So this soil is full of spores. So these two groups of bacteria, once they sense that their environment is deteriorating, they're running out of food, running out of water, or the environment is becoming polluted and toxic, and they sense that they're going to die soon, they begin the process of sporulation to make a spore. So the DNA gets replicated, there's a new chromosome, and then layer upon layer of cell wall or spore coats get deposited around that chromosome. And then you have an endospore. It's inside of that cell. Eventually the cell dies and breaks apart and releases a spore, and it's called a free spore. And the free spore just sits there and waits until favorable conditions return, at which point it will germinate and form a brand new living cell. So there are some pathogens which form spores, and we'll talk about those later on in the course, like Clostridium tetani, which causes tetanus, or Clostridium difficile, which is C. diff. And so those organisms produce spores. Bacillus is an excellent spore former, and all the members of the genus Bacillus make spores. Now, eukaryotes have some similar kinds of things. Fungi produce spores, but fungi produce a lot of spores because they're trying to reproduce. A prokaryotic cell produces only one spore because it's trying just to survive. But fungi produce a lot of spores. And certain protozoa form cysts, which are a dormant protective structure. It could be a normal part of their life cycle, or it could be in response to environmental stress. And again, they're sensing they're going to die soon. So a good example is the organism Bacillus anthracis. Any idea what disease is caused by Bacillus anthracis?
Anthrax.
Anthrax, right? Anthrax. So anthrax is a disease which can occur in the skin, right? You can get cutaneous anthrax. You can eat it and get, gastrointestinal anthrax, or you can inhale it, and the anthrax spores will germinate in your lungs and cause a very serious illness and death. So, and very interestingly, harmless organisms like Bacillus cereus and Bacillus subtilis are very, very closely related to Bacillus anthracis. Although subtilis and cereus are basically harmless, anthracis is deadly. But they're very closely related. And so here we see the mechanism of sporulation, where the chromosome has been replicated, cell wall material, that spore coat gets laid down, the endospore is formed, and the cell dies and releases that spore. And it's just dormant and waits. Now, anthrax, I know you guys are too young, but not long after 9/11 happened, there was some crazy guy sending anthrax spores through the mail. And so he had discovered a way to create the anthrax and the spores. And they do believe, I think he was a scientist up at Fort Detrick, where they do anthrax research. So there is anthrax up in Frederick, Maryland, in Fort Detrick. But whoever it was, and it most likely was this guy, because I think he committed suicide when he was discovered, but he was sending anthrax spores in envelopes. And when people opened those envelopes, the spores got released. And some of these were sent to journalists and other people like that, some politicians. But the problem with spores is that, one, they're microscopic, and two, they can survive for years or decades or centuries. And so buildings, postal facilities were potentially contaminated with anthrax. I mean, every little nook and cranny everywhere in that building, in that office, in the furniture, in the walls, in the ventilation system, there could be anthrax spores. And so the question is, well, what do we do? Because no one can inhabit those buildings because of the potential risk. And in fact, for a long time, one of the research buildings up at Fort Detrick had been sealed off and unused because it was so heavily contaminated with anthrax spores, and they didn't know how to, disinfect an entire building. And they couldn't demolish it because when you destroy it, you're simply releasing those spores into the environment. And so they just sealed up that room. So now they were faced with the problem. What do you do with these postal facilities? What do you do with these office buildings? Or do you simply close them down, for years or decades and not use them? It turns out they did find a way to thoroughly disinfect an entire building. And that was to surround it with a tent and gas it with chlorine dioxide gas. It turns out that chlorine dioxide gas will kill spores. And since it's a gas, it can diffuse everywhere, every little nook and cranny, every space, and it can get every spore. And so they were able to save those buildings, and so people could use them. Fortunately, we also have antibiotics, which are very effective at treating anthrax. So those people who did get infected survived because they were treatable with antibiotics. But anthrax is an organism that infects large hooved animals like buffalo, moose, elk, those kinds of things. So wherever you have large herds of those animals, at least where they used to occur naturally, there are anthrax spores in the soil. They're there. They're going to be there a very long time. It's just part of the environment. You don't have to worry about that around here, but maybe out west or in Alaska or in parts of Canada maybe, where you have those large herds of animals still. But anthrax was a very scary thing that was used for bioterrorism. Fortunately, we can disinfect buildings and we can treat people with antibiotics. But spores are tough, and they will persist for a long, long period of time. So, questions or comments?